Okay, we're going to look at some exam practice questions. So this is the first question in the back of your book. So this is on page 11. It shows us a length of DNA. So it says figure 3 represents a length of DNA and its cutting sites for the restriction enzymes SPE1, EcoR1, BGL2, and HIND3. Okay, so we've got 4 different types of restriction enzymes. You may want to use different colours to go through this, however you wish to deal with it. So let's have a look and see how many times. So I can see that SPE1 here cuts it once. I can see that BGL2 cuts it twice. I can see that EcoR1 cuts it 3 times. And I can see that HIND3 is going to cut it twice. Okay, so thinking about it, it's a length of DNA, so it's a linear piece of DNA. Let's go through our answers. So incubation of this length of DNA in a tube, so that just means leaving it in a tube, maybe letting it reproduce if you put in your stuff for PCR. But essentially, if you put SP1 only in there, it would result in 3 pieces of DNA. So if SP1 is cutting here, I've got 1 piece of DNA and I've got another piece of DNA. So no, that's wrong because it should be 2 pieces of DNA. The next one down is we look at it and we go, okay, HIND3 would result in 5 pieces of DNA. So let's just cut down. So HIND3 cuts here. We get 1 length, 2 lengths, 3 lengths. So HIND3 is not going to result in 2 pieces of DNA. So that one's wrong. SPE and EcoR1 would result in 5 pieces of DNA. So what I'm going to do is I'm just going to remove my markings here to make it a bit easier for you to see. If I cut it SP1 and EcoR1, I cut 4 times. So I get 1, 2, 3, 4, 5 pieces of DNA. So that works at the moment. Let's just check the last one. What would happen if I cut it at BGL2 and HIND3? 1, 2, 3, 4. Oh, hang on. There's another cut there. 5. So no, it's going to result in 5 pieces of DNA. So that one's wrong. So my answer is C. So you can see how you need to apply your knowledge of restriction enzymes, how many fragments you get from it, and so on. So the next one is a bit of an application of these. And so let's have a look. Question number 5. There were 3 suspects in an assault case. A forensic scientist found blood other than the victims at the site. The DNA was extracted from 5 blood samples. So we have the victim, the blood at the assault sites, so not the victims. So what does that tell me? That tells me that this is the suspect's blood, and then we have 3 suspects. PCR was used on the extracted DNA. A DNA polymerase enzyme is involved in the PCR process. Explain the role of the polymerase enzyme in PCR. So I want you to pause. I want you to think 2 marks, 2 points. Let's see how we go. Don't cheat. All right. Hopefully, we looked at 2 facts. So the first fact that we are looking for is that we're getting amplification, or I like this one, multiple copies of DNA. So the polymerase enzyme is going to give us multiple copies of DNA. And how is it going to do that? You could have written this in any order. The second part of that question is going to be, it's going to extend from the primer using the original DNA as a template. Some students incorrectly identified the enzyme and discussed the role of another enzyme. That's bad. You don't want to talk about ligase or or restriction endonucleases here. Many other responses gave one part of the answer. You need to look at the number of marks in order to see how detailed you need to go. Okay, so one of the regions used in the forensic analysis was a short tandem repeat or STR. Oh, yep, there we go. Sequence of 4 bases called HUM-01. This sequence, located on chromosome 11, has many alleles which differ from each other by the number of times the sequence AATG is repeated. It was this region that was amplified using PCR. The amplified samples were loaded onto a gel, and electrophoresis was performed to separate the fragments. Name 2 properties of the DNA fragments which allow them to be separated from each other during gel electrophoresis. So I'm hoping you haven't been tripped up by all that information at the beginning and just focus on the question. Give it a think, write down an answer, ready to go. Okay, we were looking for charge, size. Yep. You could also talk about, so this is the property of the DNA fragment. You cannot talk about the agar viscosity here. You cannot talk about the voltage because either of those are not properties of the DNA fragment, are they? The only thing that is a property of the DNA fragment is its charge and its size. Then we're just going to finish off this question. We're going to go down here and we're going to say, why is there only 1 band in lane 2 but 2 bands in lane 3, 4, 5, 6? Okay, this one is actually a bit of an old study design question, so I'm going to help you out with that. What we're looking for here is we're looking for the fact that in lane number 2, so these guys here are actually going to be homozygous. If you remember from your genetics further back, so you don't need to know this question, but I like to go through it anyway. So it's homozygous from way back in genetics. It means they have 2 copies of the same allele or same form of the gene, whereas all the rest of them must be heterozygous. So how many different alleles are represented? So essentially I want to know how many different positions can there possibly be for this particular gene. So let's have a look. We can see that there's 1 here, 2, 3, 4, 5. So there should be 5 alleles because there are 5 different possible spots for that to end up. Now, which piece of DNA, A or B, has the greater number? So again, I don't think you're really going to need to answer these because they are to do with genetics, but I like to go through them because I think you can answer those ones. Which piece of DNA, A or B, has the greater number? You should be saying A because it's closer to the top. And which suspects or suspects seem to have committed the assault? So remember, lane number 3 is the victim, is the other blood, so that's the suspects, and the one that matches it is suspect 5. Okay, and that's what we were looking for. So the gel, we said back here, why is there only 1 band in lane 2? We said that was because it was homozygous, 2 copies of the same length. These guys were heterozygous because they had 2 different lengthed fragments. We said 5 alleles. We did talk about that because there were 5 down there. Which one, A or B? We said A. So 5A, 5A, and suspect 5 down here as well. So they were all correct. And so that was just one question. It's a very old study design question. There's some things that are missing. It's actually the exam that I sat at the end of my year 12. But that should give us an idea of how to approach a question like that.